So with the help of this video, we are going to learn about natural selection, phenotypes and genotypes. So in this chapter, we examine some of the major consequences of natural selection for the species under selection and for other species with which they interact. Selection on one species can influence the evolution of other species through evolutionary processes such as mimicry and co-evolution. Selection acts on the fitness differences among different phenotypic traits of individuals within populations. So selective changes is not unbounded, however. Selection is facilitated by the large amount of allele variation or polymorphism in populations, variation itself being subject to selection. Response to selection is influenced by constraints, channelization, the ability to produce the same phenotype in response to environmental or genotypic variation, the plasticity arising from genetic and developmental processes. How available genetic polymorphism responds to changing environments and phenotypic plasticity, mold, selection, and influence fitness are discussed. So let's have an introduction about this topic. Selection acts on the fitness differences among different phenotypic traits of individuals within populations. So selection acts on the fitness difference between or among different phenotypic traits of individuals within a population. Species are bound together as potential competitors, as predators and prey, through mutualistic associations such as mimicry, and as communities through ecological interactions. So consequently, selection on one species influence other species. However, selection is not limitless. Dynamics and constraints of genetic and embryologic developmental pathways work with selection both to provide plasticity and to narrow or canalize evolutionary change. The interaction can be observed in nature and mimicked in laboratory experiments in which an organism's response to environmental change is measured as a reaction norm. The greater the number of reaction norms, the more responsive the organism is to environment. Then selection on features of one species can affect other species. Species compete with each other for resources and through competition in ways that affect fitness. Species also co-evolve and adapt an advantage or increase in fitness for one species may bring about deterioration in the environment for other species and a potential decline in their fitness, or it may enhance the fitness of the other species. In 1976, American evolutionary biologist Leinwald Wallen in 1935 to 2010 drew the analogy between species survival and a remark made by the Red Queen whom Alice meets in Lewis Carroll's Through the Looking Glass. Here you see it takes all the running you can do to keep in the same place. According to the Red Queen hypothesis, selection on one of two competing or co-evolving species gives that species a competitive advantage, reducing the fitness of the second species and eliciting selection in that species. Co-evolution happens here. So co-evolving species in a changing biological environment therefore face an arms race. A race most obviously seen in the co-evolution of predator and prey. Even though the second species would seem to be at a disadvantage, the long-term consequences of the Red Queen's theory is to increase the competitive fitness of each interacting species. As Darwin stated in Origin of Species, if some of these many species become modified and improved, others will have to be improved in a corresponding degree or they will be exterminated. Next topic, selection and constraint. So for populations to evolve, that is to change their gene frequencies, mutations must introduce nucleotide differences in the alleles of individuals within the population, adding to the existing genetic variation. The mere appearance of new alleles, however, is no guarantee that they will persist or prevail over others. Explanations for the persistence of many mutations and their increase in frequency must be sought in process other than the original mutational event. The existence of developmental systems with their regulated pathways raises the question of whether development constraints or enhances through plasticity, structural and fundamental variation, thereby influencing natural selection and future evolution. For example, one can ask why insects have six legs and land vertebrates four, then almost any even number of legs or even none at all can suffice for locomotion. What role, if any, do the dynamics of development play in maintaining such phylogenetic constraints as the number of legs in insects and invertebrates? And how do such mechanisms relate to natural selection? The answer is not entirely simple, although development, on the other hand, limits the range of forms that natural selection can bring forth, and on the other hand, provides plasticity for changes in the phenotype. Although many developmental changes are possible, certain of these changes, especially those of large degree, are mostly lethal. Their effect cannot be successfully integrated with other aspects of embryonic development. The need for integration, generation after generation, results in developmental and phylogenetic constraints. So, in this picture, I could see the number of neck vertebrae in mammals is conserved at 7 in most mammals, shown here in two species with very different lifestyle, a whale and giraffe. But their neck vertebrae, like cervical vertebrae, is conserved in at 7. So variation can occur, however, as shown in the three-toed sloth, which has 8 or 9, and the manat, which has between 5 and 7 neck vertebrae. So there are differences. A constraint is any influence that acts to canalize or channel the effects of selection. An example of a constrained feature is the presence of 7 neck vertebrae in practically all mammals, whether short necked whales or long necked giraffes. In order to understand evolutionary conservation or change of phenotypes, the process that underlies constraint must be integrated with the role of natural selection. Constraints and selection interact to channelize evolutionary change. A comparison between evolutionary change in frogs and placental mammals illustrates differences in evolvability of phenotypes, reflecting as yet unknown differences in constraints on evolution between lineages. Frogs appear in Triassic deposits about many years ago. Placental mammals do not appear until the Cretaceous about many years ago. So, despite their much longer fossil history and their large number of species, frogs have undergone fewer phenotypic changes in evolution compared to the enormous adaptive radiations of placental mammals. The more than 3,000 species of frogs are so much alike that herpetologists place them in a single clade, the order Anura. The 4,300 species of placental mammals, of which about 2,000 are rodents, diverged widely and usually are classified into 18 lineages or orders ranging from bats to primates to whales. Later, previous selection history and networks of developmental genetic interactions channelize subsequent evolution. So, compatibility of new phenotypic features with established genetic network channels selection. Furthermore, selection itself can act as a constraint. The potential to respond to subsequent selection regimes may be compromises by past selection. Selection toward becoming a master of one trait prevents an organism from becoming a jack of all traits, and vice versa. As one of the many examples, selection for increased efficiency of swimming performance impacts traits that enhance running on land, as seen in crocodiles. For example, maintaining phenotypes that can successfully face all possible future eventualities is unattainable in a single lineage. Selection sees what is current, not what sees what might be optimal for an unknown future. So, when we look at the molecular tree of mammalian relationships, phylogenetic tree of placental mammals, monotremes are there, marsupials, then tubulidentata, Afrosauricida, Macrocelida, Proboscida, then Hydrozoidea, and so on. And along with that, primates, Rodentia, Carnivora, Pholiodota, and Chiroptera, Perissodactyla, etc. So, nevertheless, constraints are not impossible barriers. Some mammals escape the constraint of seven cervical vertebrae, like three toed sloths have eight or nine, and mantis have six. Given sufficient time and new opportunities, vertebrates lineages that evolved from swimming fish into crawling terrestrial forms later evolved into different marine swimming forms like ichthyosaurus or whales, and into entirely novel winged aerial forms like birds, bats, and pterosaurs. Therefore, developmental constraint narrows variation but does not prevent evolutionary change, illustrating the importance of understanding exactly how the limits to variation are set. So, then selection, changing environments, and plasticity. When exposed to altered environmental conditions, some species respond by producing more than one phenotype. This ability goes under various names, including phenotypic plasticity. According to terminology introduced early in the development of Mendelian genetics, the range of phenotypic expression that can be produced by a single genotype in response to environmental change can be expressed as a reaction norm. So, reaction norms apply to phenotypes encountering different environments. They can be compared for the same species under different environmental conditions or for different species under the same conditions, which was explained with the help of this figure. So, reaction norms are detected experimentally by raising individuals in different environments, different temperatures, predator densities, and so forth, measuring the trait of interest under each set of conditions and plotting the phenotype against the environmental variable. A reaction norm, therefore, is an index of organismal environmental interaction on the one hand and flexibility or plasticity, channelization or constraint on the other. A single line, a single reaction norm indicates a tightly canalized response and limited responsiveness to environmental changes. Multiple slopes indicate multiple reaction norms and a plastic response of organism to their environment. So in 1966, Waddington and Robertson reported the results of experiments in which developmental channelization was selected for, like channelizing selection, and resulted in enhanced phenotypic variation. So, canalizing selection limits the expression of a trait, presumably by eliminating genotypes that would broaden phenotypic responsiveness to the environment, and to broaden expressions of the character, which would occur by eliminating genotypes that would permit an individual to respond to any genetic, epigenetic, or environmental influences that would lead to greatest variability in the phenotypic expression of the trait. So, here we could see this picture which shows the distribution and migration patterns of four congeneric species of sternicats in Europe and Africa differ. The insert shows male and female European stocknets or Saxi, Saxicolia rubicola, it's a bird, on the left side, and African type of this bird on the right. Reaction norms for age at molt against time Fourier for three species analyzed so differ, and the Siberian type of this bird shows much less plasticity in its feature than do the other two species. Among this Rattleton's experiments on channelizing react selection were demonstrations in Drosophila that phenotypically uniform expressions of the normal ultrabithorax gene in the face of environmental stress depends on other background genes. The genetic basis for such constant expression becomes apparent when specific genetic loci that support ultrabithorax transcriptional stability was identified. New features such as additional pair of wings seen in Drosophila as a result of mutations in genes such as ultrabithorax can be elicited by environmental stimuli and then selected for in the absence of the stimulus. The traits become genetically incorporated and appear in subsequent generations that have not been exposed to the environmental stimulus, an evolution process known as genetic assimilation. This is not a Lamarckian process of direct instruction by the environment. It occurs because of the presence of unexpressed genetic variation and selection for genotypes that are capable of responding to the environmental change. The close fit between organismal flexibility and environmental change is a product of underlying genetic components. This was well demonstrated by Brandl and Flat in 2006 using color polymorphism in the tomato hornworm. Presence of more than one color morph or polymorphism is regulated by juvenile hormone and is sensitive to environmental signals that influence the action of juvenile hormone. The polymorphic population of hornworms exposed to a heat shock and then selected for color morphs over multiple generations produced the environmentally sensitive color morph in the absence of further heat shock. Then selection and fitness. Although hereditary transmission is through the genotype, selection acts upon the phenotype to the extent that phenotypic differences depend upon genetic variation. Phenotypes differ in variability and fertility and thus influence the frequency of their genotype. The consequences of selection on a particular trait in population are measured in terms of reproductive success or fitness. In their simplest form, fitness and the intensity of selection are measured by the number of descendants produced by one phenotype relative to those produced by other. However, in the case of plasticity and reaction norms, there can be differences in fitness among phenotypes produced by the same genotype. Sustained selection on the phenotype can result in changes in gene frequencies from generation to generation, especially if genetic assimilation is opening. So, the effects of selection are recorded as selection coefficients, which give the greatest effect on fitness, a value of 1, which adaptive value being defined as 1s. The lowest selection coefficient would be 0, indicating no effect on fitness, as seen in mutations on third position codons, like Wobble hypothesis is there. So, third position codons change mutations would not be contributing to protein production, will not be producing protein. So, we refer to the situation where the heterozygote has superior reproductive fitness over both homozygotes as heterozygous advantage, because heterozygous advantage is often expressed in characters that affect fitness like longevity, number of offspring produced, resistance to disease. It has been studied in some detail as heterosis, and often cited example of heterosis is the dramatic increase in the agricultural yield of hybrid corn achieved by crossing selected inbreed lines. Then, beginning with an average yield of about 25 bushels per acre in 1920, selective breeding of hybrid corn enabled increase in yield to as much as 140 bushels per acre. Then, an example of the interplay of factors affecting fitness is the observation that fitness of the phenotype can be frequency dependent, especially in situations where a variant allele is either common or rare. Frequency dependent fitness allows single evolutionary events, the origin and spread of a single mutation, to be modeled and compared. Understanding frequency dependent fitness has the potential to inform us about branching points in evolution. So, too, does the approach discussed that considers selection and fitness in terms of populations occupying different adaptive peaks rather than valleys in an adaptive landscape. In this approach, gene frequencies change as the result of what is termed genetic drift. The next topic, selection and balanced polymorphism. Persistence of different genotypes to heterozygote advantage is an example of balanced polymorphism, a term coined by Oxford ecologist genetist E.B. Ford in 1901 to 1988 to describe the preservation of genetic variation through selection. A gene locus is defined operationally as being polymorphic if at least two alleles are present, and if the second allele or the least frequent allele when more than two alleles are present has a frequency of at least 1%. Polymorphisms are ubiquitous in practically all populations examined. One well-known example of polymorphism is the sickle cell gene in humans, in which the sickle cell homozygote is selected against because it cannot carry oxygen efficiently. Because individuals who are heterozygous at the sickle cell locus are more likely to survive the malarial parasite than are those who are homozygous for either of the two alleles. Homozygotes remain in the population. This gene and others shown with the help of this picture of protection against malaria persist in notable frequencies in geographical areas where malaria is endemic. Of course, while retaining the ability to induce sickle cell anemia, the malarial parasite has evolved multiple genes to evade the immune response mounted by the host. Individual Plasmodium falciparum parasite contains some 60 genes for one protein involved in transmission. Different individuals contain different variants of the genes, and impressive arsenal mounted by the parasite in the parasite host arms race. A second example is the phenotypic level as selection of cryptic moth prey by blue jays like some other Cyanocita cristata. Jays capture and consume more of the abundant prey types than of the rare types, switching capture strategy if the rare prey type becomes more abundant. As a third example, selection and balanced polymorphism can be demonstrated in laboratory experiments in two types of E. coli are co-cultured in glucose-limited minimal media. The, the relative abundance of each is frequently dependent, each type having an advantage when rare, resulting in maintenance of a balanced polymorphism in the population over time. An influential alternative theory to selection and balanced polymorphism was proposed by Moto Kimura in 1924 to 1994, in 1979, and expanded into a book in 1983. The the naturalist hypothesis, which was discussed briefly with the help of next picture. I will be explaining that topic. So, prompted much controversy that led to research and development of evolutionary theory. So, what is that naturalist hypothesis? Kimura in 1979 to 1983 argued that the rate and amount of evolution at the molecular level is far more rapid and more pervasive than had been appreciated. Consequently, he proposed that not all polymorphisms results from selection, asserting that the presumed high cost of selection necessitated that most amino acids changes be natural, that is, that they have no effect on the phenotype. Kimura argued that as selection does not affect on natural mutations, the fixation of such alleles incurs no genetic cost and depends only on their mutation rates and on random changes in gene frequency, like genetic drift. This proposition becomes known as the naturalist hypothesis. Neutralist. Sorry for mispronouncing. Neutralist hypothesis, although it was called non-Darwinian evolution by some because of its dependence on mutation and not on selection. The extensive enzyme and protein polymorphisms in natural populations, demonstrating that multiple alleles exist at many thousands of loci in many species, seemed to support the neutral hypothesis. High frequencies of selectively neutral mutations in rapidly mutating strains of E. coli provide one line of support for the neutralist position. Moreover, and in contrast to the neutralist hypothesis, recent extensive studies have demonstrated functional consequences of polymorphism. From a survey of insects, mollusks, vertebrates, and plants from Israel, it was concluded that the amount of allozyme polymorphism in a species correlate to factors such as life, habit, and climate. Even more striking is the finding that clones of the bacterium E. coli isolated from the intestinal tracts of animals as diverse as lizards and humans, and from localities as widespread as New Guinea and Lua, share common allozyme frequencies. For each of the five different enzymes, one particular allele is present in almost all samples. As other allozymes of these proteins exist in E. coli, such as narrow distribution of allozymes, is difficult to explain on any basis other than selection on a favorable allozyme. We know now that mutations in coding genes are strongly selected against and that many mutations occur in a large amount of non-coding DNA, much of which is involved in gene regulation. So nucleotide sequence analysis show that polymorphisms are significantly more frequent in those DNA sequences that do not determine amino acid sequence than in those that do. For a given regions of the genome, more polymorphisms occur in introns like intervening sequences that do code for amino acids than in exons like expressed sequences which do. The alcohol dehydrogenase gene in Drosophila melanogaster has 6% polymorphism in introns, 7% polymorphism in third position codons, and almost no polymorphism in exons. Because random events can hardly explain such pronounced differences in polymorphism. Such findings strongly suggest that selection must be discriminating agent that determines which nucleotide based substitutions will become established in functionally different DNA sequences. So, next topic, selection and mimicry. Other conditions responsible for polymorphism include a change in selection coefficients, reflecting that gene detrimental at one time are advantageous at another. Selection against a gene may depend on its frequency and may change or even be reversed when the gene is at low frequency and before it can be eliminated. So, an example of such selection is Batesian mimicry, a mechanism proposed by explorer Henry Bates in which palatable species that mimic distasteful models are protected against predators. So, in this figure, we have explained about the case of about malaria, which we explained earlier. So, falciparum malaria, sickle cell anemia, beta thalassemia, then G6PD deficiency, palladium deficiency. So, relationship between the geographic distributions of the most dangerous form of malaria, falciparum malaria, initiated by the protozoan parasite Plasmodium falciparum, and genes that confer resistance against malaria. Then distribution of falciparum malaria in Eurasia and Africa before 1930. Then distribution of the gene for sickle cell anemia. Then distribution of the gene for beta thalassemia. Then D. The picture D shows distribution of the sex-linked gene for glucose 6-phosphate dehydrogenase deficiency, not palladium. This glucose 6-phosphate dehydrogenase deficiency in males is in frequency is about 2%. So these are shown with the help of this figure. So mimicry in general, the most frequent the mimic and the less frequent is its model, the greater the chance that predators will attack the mimic. Conversely, the less frequent the mimic compared to the model, the greater the chance that the mimic will be protected. So, mimicry in insects evolved early, and as expected, if mimicry is effective, has been remarkably stable. And you've seen leaf mimicking stick insect, Eophilium mesenlysis from 47 closely resembles extant insect mimics, which is shown in this figure, and so is interpreted as a mimic rather than as an example of camouflage not involving mimicry. Furthermore, this ancient insect mimicked the leaves from several types of plants, including myrtles and laurel trees and alfalfa, indicating the sophisticated forms of mimicry were already present in the Eocene. So when we look at this picture, we could see this Eocene stick insect, which we just mentioned, is a leaf mimic, bears a strong resemblance to extant leaf mimicking stick insect. As shown in this picture, mimicry between different species like Mullerian mimicry benefits both species by enabling predators to learn a single warning pattern that applies to all the potential but distasteful prey. When rare conspicuous warning patterns on unpalatable individuals offer little protection, predators have few chances to learn their distastefulness. Then, distinctive patterns, however, offer greater protection to unpalatable individuals when they are at higher densities, as shown by a Texan grasshopper that develops color that blend in with the vegetation when individuals develop alone. When the grasshoppers develop in groups, however, they develop yellow and black warning colors if they consume a plant that is toxic to predators, providing a further example of phenotypic plasticity. A particularly detailed analysis of the evolution of mimicry concerns the black Sella lily, Arum palinate palestinum, sorry, that attracts fruit flies Drosophila for pollination by producing chemicals that mimic odors produced by yeast. Fruit flies have evolved a set of odor receptors that detect volatile chemicals released by yeast, a major part of the fruit fly diet. Of eight Drosophila species examined, seven share the same olfactory response to the volatile chemicals, despite 40 million years of independent evolution separating species pairs. So this picture shows mimicry in different species of butterflies. Batesian mimicry by a North American species in which the more palatable viscerally butterfly right mimics the more unpalatable monarch left. Resemblance between the two South American unpalatable species. And picture B shows common warning pattern to predators and helps protect both prey species like Mullerian mimicry. Mimicry is one of the most obvious example of convergent evolution. So mimicry is one of the most obvious example of convergent evolution. Then selection in changing environments, melanism. Next topic, selection in changing environments, melanism. A population sufficiently widespread to occupy many environments may maintain a variety of phenotypes, each of which is superior in a particular habitat. Spatial and temporal organization of the environment may significantly affect the extent to which population will rely on genetic polymorphism as an adaptive strategy. Coarse-grained environments in which different individuals in a population are exposed to different experiences promote greater genetic polymorphism than do fine-grained environments in which all individuals experience relatively similar conditions. A prominent example is polymorphism associated with industrial melanism. And in industrial areas of England in the 19th century, vegetation was darkened by deposition of soot from the burning of coal and by exposure of the underlying bark when the soot killed off surface lichens. First observed in Manchester and later spreading to other areas, population of certain moths and butterflies were found to have increased frequencies of dark-colored melanic morph. In the early 1970s, the industrial city of Birmingham, Bernard Kettlewell in 1910 to 1979 and other released no numbers of light and melanic morphs of the British peppered moth like Biston betullaria. A significantly greater proportion of melanic than light forms were recaptured at a later date. So Kettlewell's data suggested that areas covered in soot offer greater protection to melanic than to light-colored moths. The light forms are at a selective disadvantage. Thus, more melanic than light moths survive to be recaptured. The relative fitness of the melanic moths may lie in at least partly in their ability to remain concealed from bird predators on darkened twigs or tree trunks, which can be explained with the help of this figure. Like dark colored and light colored tree trunks are shown, one with a melanic and one with a non-melanic, non-melanic peppered moth placed to show the contrast between them. First one is not that much visible, but the second one is clearly visible as background is black, but that particular moth is light shaded, so it's easily visible. That causes disadvantage for that one. The light colored trunks derive their appearance from lichens, although trees are commonly shown as resting sites for these moths. Their actual resting habits are unknown. So, in non-industrial areas, in contrast, trees covered with normal gray lichens offer decided advantages to the light-colored moths. Whether via environmental camouflage or other unidentified factors, English populations of the peppered moth show various degrees of polymorphism ranging from high frequencies of the melanic form in industrial areas to almost zero melanic forms in many rural areas. Researchers were able to determine how long it took the frequency of non-melanic moths to decrease in industrial areas using the dates when specimens were collected by amateurs and records in museum collections. Assuming one generation per year, they eliminated it took about 40 generations for the frequency of non-melanic phenotypes to decrease from 98 to 5%. The selection coefficient 0.2 or even higher is indicative of intense selection against a non-melanic gene in this industrial areas. Hmm. Passage of clean air legislation in British in 1956 reduced industrial smoke in many formerly polluted areas. This reduction in pollution altered selection, and the frequency of melanic forms of the peppered moth and other insects declined dramatically. The difference between the light colored and the melanic forms of British peppered moth is now known to be due to allelic differences at the single locus carbonaria recently identified by genetic mapping. Crossbreeding with the melanic form of the moth found in the United States demonstrated that the same locus is involved in those widely separated populations. A parallel situation is seen in light and dark forms of the rock pocket mouse, which is shown with the help of this picture, found in the American Southwest. Mice with light sandy colored fur lacking melanin granules occur on sandy substrates. Mice with dark melanin containing fur are found on black lava flows. These differences in coloration reduce predation from birds and mammals that are visual hunters. Ability to maintain the two fur colors has been traced to the gene MC1R that codes for the melanocortin 1 receptor. MC1R plays a key role in whether melanin granules will be deposited into the fur in rock pocket mice and in many other animals. Rock pocket mice is shown that forage on sandy substrates have sandy colored fur, while those that forage on black lava have black fur. So mutations also underlie the melanic form of the beach mouse, which is shown in the southeastern United States. Four amino acid differences separate the MC1R gene in light and dark rock pocket mice. A single amino acid substitution in the bleach mice from Florida is associated with an adaptation to its changing environment. In contrast, 63 differences separate wild house mice indigenous to Europe from rock pocket mice. We could conclude that small genetic changes can lead to significant phenotypic changes, illustrating selection and evolution in action. Furthermore, the pathways regulating the deposition of melanin granules lie at the basis of color change in other species and not only rodents. Mutations in MC1R are associated with changes to pale coloration in several species of wild cats, lizards, and snow goose, Arctic skua, and Kermode black bear, all of which also have melanic forms. Pale forms of two species of lizards that live on white gypsum dunes of white sands formation in New Mexico result from mutations in MC1R. However, the origins of their coloration are different. In one species, it is traced to mutation in receptor signaling, in the other, to integration of the receptor into melanocyte membrane, some gene different mechanism for loss melanin. Similar phenotypes can arise by evolutionary modification in different aspects of the function of a single gene or different aspects of the functions of different genes.